Okay, so now I'm going to introduce our third speaker, who is a second keynote speaker in this session, and this is the last talk in this session. So the speaker is Professor Rong Fan. He is the Harold Hodgkinson Professor of Biomedical Engineering at Yale University, and also a professor of pathology at Yale School of Medicine. He received PhD in chemistry from UC Berkeley and completed his postdoc at Caltech before joining Yale as a faculty. His lab has been developing many cutting-edge spatial omics technologies, including dBit-seq for spatial transcriptomics and an extension of it to the multi-omics, epigenomics. That's really innovative technologies. He also co-founded several companies, including Isoplexis, Single-Run Biotechnologies, and Atlas Omics. He is the recipient of a number of awards, including National Cancer Institute's Howard Termin Career Award, Career Transition Award, excuse me, and also the NSF Career Award and the Packard Fellowship for Science and Engineering. And he's been also elected to the American Institute of Medical and Biological Engineering, the Connecticut Academy of Science and Engineering, and also the National Academy of Inventors. So thank you so much, Ron, for joining us today, and we are all looking forward to your talk. Thank you.
Yeah, thank you. Thank you, Tetsuya, for a very kind introduction, and thanks the Plant Cell Atlas Consortium for having me. So I'm gonna go ahead and share my screen first. So can you see my screen?
Yes.
Yeah, it's such a great honor since I have now been working on plant biology in my laboratory. So I would like to kind of give you a little bit overview of the spatial omics technology landscape and also what the future might look like in terms of technology and what we are continuing to develop in my laboratory. And all right, so this is my disclaimer. And so I have been using this slide. This is probably the only slide showing real plant biology. I have been talking about this slide over and over in my previous talks. It's such a great pleasure. Finally, I can speak to the plant biology audience. And so honestly, when the microscope was discovered by Robert Hooke back to many hundreds of years ago, and if you look at the image captured by the very first microscope, you'll probably already realize actually the cells, for example, here and there, it is in the same plant leaf, actually look different. So nowadays we know the cells are, their functions are already determined by their kind of genetic, transcriptional, and proteomic programs. And you might guess, okay, they should have different omic features underlying the different morphology and the different function location of the cell. So sort of from the very beginning of the cell biology and discovered by the invention of the microscope, we already believe the biology needs to be spatial. So now I'm gonna jump to the technology landscape. I used to call this technology kind of landscape or sort of the current state of our technologies, but now I kind of try to use the word technology family because oftentimes we need all kinds of technologies and combine and integrate the different data sets to really get a complete picture about the question you want to ask. So broadly speaking, I think over the past 10 plus years, there are two major directions of technology development to achieve spatially resolved high-flex protein or gene expression analysis. One is based on imaging, the other is based on kind of high-throughput NGS sequencing. And so in the imaging-based approach, I was truly amazed by the accomplishment done by our microscopy colleagues over the past 10 years. I think literally 10 years ago, if you can image just 4 or 5 fluorescent markers, you are very good. You are basically the best in the world doing fluorescent microscopy. But nowadays that can increase to for proteins like over 100, and for the gene expression using fish, two technologies like merfish and sigfish allow you to image almost the entire transcriptome sort of genome scale gene expression analysis. And so NGS-based approaches came out a little bit later, but over the past couple of years, I think there are kind of a lot of technologies that emerged to do unbiased genome scale gene expression analysis, but also this can be go way beyond just the gene expression. I think as demonstrated by our own laboratory, but also over the past couple of years, you can see the NGS-based spatial omics technology can achieve the resolution down to single cell or even subcellular. So I'm gonna kind of give you a little bit more details about all those different kind of representative technologies. But the field is not limited to just a panel of proteins and genes and a transcriptome, although that's sort of the majority of the technology development efforts in the space. Some technologies emerge over the past 2 to 3 years to allow you to do genome-wide, protein-wide unbiased proteomic discovery by mass spec. And another interesting aspect in the metabolism, right? Can you map down metabolic activity directly in the tissue using mass spec or using some microscopy technologies? And now you can see the technologies emerging over the past couple of years can really sort of capture not just the spatial, but also different layers of the omics, spanning from genome, transcriptome, and proteins on the way to the function of individual cells, such as metabolic activity. And so some historic events, I try not to, talk about the full history. Actually, it's a beautiful review article written by Dr. Leo Patcher called the Museum of Spatial Transcriptomics. And if you read that paper, you'll realize actually some early developments such as the single molecule fish really kind of paved the road towards the modern spatial transcriptomics, imaging-based transcriptomic technologies. And I would like to highlight two of those. I think one is the single molecule fish for the first time demonstrated by Rob Singer back to actually many decades ago. And so that allows you to really look at RNA biology in situ. So I'm probably a little bit conservative. I would not call that omics. I would not call that transcriptomics. But that allows you to probe the biology and RNA biology in situ. But over the past kind of less than 10 years, including merfish and a six scope and sigfish, I think the key innovation here is you're able to do combinatorial barcoding rather than you just image each transcript at a time, and that combinatorial encoding mechanism allows you to get the total number of genes to image up to entire transcriptome. So I would say this is not just a linear evolution of the technology, but what happened during the past 10 years is a really kind of major leap driven by this complete new concept that lead to the omics level imaging of gene expression. So technologies I kind of mentioned in a technology landscape slide for imaging based, you can image either proteins or just the transcripts. Or for imaging proteins, oftentimes you can combine with limited number of transcripts. And so very early on, I think based on sort of the mass cytometry, imaging based on flow mass cytometry, you can tweak to turn that into imaging mass cytometry developed by Professor Gary Nolan. There are two methods to do that. They are very, very similar downstream, but it depends on how you kind of ionize the metal probes using either laser beam or use the ion beam. The two technologies now commercialized by two different companies called either MIBI or IMC. So in this case, you don't use fluorescent probes. Instead, you use the kind of rare earth metal probes to label the proteins of interest, or you can label other molecules as well. And the radar is done by mass spec, by spot to over entire surface of the tissue. But you can still go back to use fluorescence. But we all know we're limited by sort of how many fluorophore we can choose, and also limited by the spectral overlap and within the UV-Vis wavelength range. So in that case, single imaging cycle cannot give you extremely high plex, but you can do repeated kind of imaging and a removal of the fluorophore and over many cycles, you can get up to nowadays demonstrated by Akoya Biosciences up to 101 protein markers. If you're interested, there's a paper published in Gene Biotechnology last year that really set the world record in terms of multiplex immunofluorescence imaging. So I think NanoString came up with a similar approach to look at both gene expression, but also panel proteins. But I think how you design a probe, that's quite different. But the fundamental principle is you do single molecule imaging using some sort of amplification here in this case. And you can get enough optical intensity to image individual molecules. So that has been commercialized as early on called a spatial molecular imager, but now it's called a CosMx. And other methods based on imaging, but it's called a sequencing, called in situ sequencing. And if you label the gene you are interested in using this sort of padlock probe, and then do rolling circle amplification to give enough signal you can read out by single molecule imaging, and you can sort of directly image individual transcripts in the tissue by sequencing this kind of padlock probe that bind to the genes of interest. And so in this case, I think a small number of nucleotides within that sequence allows you to really increase the number of transcripts you can detect, because this combination of different the different sort of the nucleotides in the region you want to sequence can give rise to a very large number of genes you can detect. So this is in situ sequencing, and very similar technology called PhysIC, and both technologies have been sort of acquired by 10x Genomics. Now that's commercialized system called Xenium. So I already talked about the MERFISH developed by Professor Xiaowei Rong. So that's also commercially available by Visgene. It's called a MERSCOPE system based on the single molecule fish, and that's truly single molecule fish. You don't need to do amplification. All right. So I think I talk about the imaging-based spatial omics and largely spatial transcriptomics, and you can also image small panel proteins. But what sort of emerged over the past couple of years, literally less than 10 years, and actually a major breakthrough kind of took off to 2019, only 5 years ago. The landmark paper published by Professor Joachim Lomberg back to 2016 demonstrate you can capture messenger RNAs over the surface of the DNA microarray. And if you put in the tissue section on top of the DNA microarray, you'll permeabilize the tissue and capture transcripts that diffuse from the tissue to the surface of the DNA microarray. But each DNA microarray spot has unique DNA sequence that determines the spatial address code. And when you do a cDNA synthesis, you're going to incorporate that spatial address code into the cDNA. And such that when you collect all cDNA and the sequence, you can go back to see where that cDNA molecule is coming from based on the spatial address code. So the same fundamental concept has been sort of utilized by many different technologies, and they continue to push the the limit of sort of the boundary of the whole field by reducing the spot size such that you can get down to I think back to 2019 or like a 10 micron each. But over the past 2 years, you see field technologies that can get down to 1 micron, even sub-micron. And just a couple of months ago, super exciting. And 10x Genomics Visium HD commercially launched. You can get it down to truly kind of cellular or subcellular resolution, but they all follow and the same fundamental principle. Professor Joachim Lamberg coined called a barcode solid surface for MRI capture and spatial transcriptomics. So now I'm gonna kind of move to what we are working on in my laboratory. So we are also interested in NGS-based spatial transcriptomics and the spatial omics, broadly speaking, because that gives you unbiased global profiling capabilities of the truly unbiased discovery power to look into the mechanism and the whole genome scale, right? Genome-wide mechanistic study. And so, but the approach is kind of totally opposite. Instead of taking messenger RNAs out of the tissue and capture on the barcode solid surface, we send the DNA barcodes into the tissue to barcode either transcripts or panel proteins, and later we found you can barcode many different things, including epigenetic features, including the different RNA species. And later, I think other approaches, including SlideSeq, they also use this in-tissue barcoding rather than just you capture over the surface of the beads. So it seems like it's extremely versatile, and it does represent a whole new direction in the field of spatial omics. So when we kind of published that paper to show spatial sort of the whole transcriptome plus a panel of proteins, we only profile a panel of about 32 proteins. And honestly, I would not call that proteome, right? But the question is, can we really map the hundreds of proteins, which is totally possible, turns out. And we were able to develop a panel of antibody DNA conjugates to map about 300 proteins for a variety of human tissue samples, about 200 for different mouse tissue samples. And so this is all about the targeted kind of protein detection. But once you have the panel extremely large, we found even using the proteins only, and the different proteins give you kind of complementary information, and you can do much better. So it's clustering based on the proteins to identify different tissue features, different cell types directly in the tissue. All right. So as I said, we were not just trying to develop another spatial transcriptome technology from even the very beginning. I believe this spatial barcoding, in-tissue barcoding approach is very versatile. You can look at many other things. What we are excited about for many years is the underlying mechanism that controls the gene expression, for example, epigenetic regulations, right? There are many different epigenetic regulations out here, and we decided to start with two of those, okay? Specific histone modifications, or the global accessibility of the chromatin state. All right. For specific histone modification analysis, so we took the beautiful chemistry called a cut and tag, which utilize the antibody that can recognize the specific histone modification, and then the protein A, the TN5 transposase can recognize the specific antibodies against the histone mark. And then once you activate the TN5 transposase, you're going to cleave the DNA, genomic DNA, and a loci associated with the specific histone modifications. After your sequence, you get on a pixels spot by spot across the entire tissue, and then you perform unsupervised clustering. So for different histone modifications, we we got this unsupervised clustering. You see the different clusters. You can go back to see where the different clusters are located. That's the beauty of this spatial. Unlike single cells, you just get a bunch of those clusters. But in our case, we know where they are coming from, right? But without looking at how sort of the multiplex histone modification analysis across the adjacent or several tissue sections, you can already appreciate so different histone modifications control the different tissue types in different regions at this particular stage of the mouse embryo. For example, right here, the K4 trimethyl doesn't really resolve that much between here and here, but the other histone mark K27 trimethyl allows you to resolve other features. I would not say to resolve, though basically that tells you this histone modification does play a role in the development of those the tissue or cell types. This histone modification doesn't really cannot play a key role over here. But you can see that in this particular region, there's the heart. You can even see in the heart chamber, and there's a atrium, there's a ventricle, and so it seems like this histone modification plays a very important role in the heart development at this particular stage, but not this histone marker, right? So people constantly ask me these questions. Okay, the histone modification cut tag versus A tag versus gene expression, which one does better job to determine cell type? I really think that's not a the perfect relevant question. And the question should be, in our case, I really think the spatial cut tag or spatial histone modification sequencing tells you biology, tells you whether or not this histone modification actually does something to the development of this particular tissue type or cell type in the mouse embryo. So that's the biology, not just the cell type or cell identity. So once you perform on a clustering, you can go back to see where they are and the different major clusters showing here. So you appreciate actually very distinct neuronal cell types in different brain regions. You can resolve very nicely, and each cluster you can see the genome trace, right? You can see the peaks corresponding to all different genes. And so in this case, keep in mind this is a genome-wide sequencing data. You can look at whatever genes you are potentially interested in. You can visualize those individual genes as well, but in this case, this looks like a gene expression, but no. This tells you the gene activity score associated with this specific chemical modification of the histone, K4 trimethyl at histone 3. All right, so this is sort of, you can now direct the visualize biology rather than just showing what are the different cell types where they are. So actually what you see is the biology associated with the different cell types. So what about the global analysis of epigenetic state using the elegant chemistry called ataxic, developed by Greenleaf and Howard Chan at Stanford. And so the TM5, it's very similar to cut tag, but just you don't use the specific antibody, and you just use the TM5 transposes. You pre-assemble with the DNA oligo. Once the transposes cleaves the open chromatin region of the genome DNA, you're gonna insert this DNA oligo into the open chromatin region. If you do bulk ataxic, you can just go ahead and lyse the tissue and the sequence. But in our case, we did 2 more steps to ligate the barcodes A and the barcodes B, and then you can have like a spot by spot or pixel by pixel sequencing of ATAC data trace. You can, for example, visualize all those pixels within the embryonic mouse liver, or you can aggregate to produce this kind of pseudo bulk embryonic mouse liver ATAC data trace. You can see this a pretty strong concordance between our data and the ENCODE reference data. So similarly, once you perform unsupervised clustering on pixels, you can go back to see where the different pixels are. So you can see that in the neuronal different neuronal cells in the brain region, you already see quite distinct clusters. And this is the spine, very thin stripe. We could not find any reference data because no one actually dissected the spine to perform epigenome sequencing, but now we see it's a very distinct cluster. And liver versus the liver here, the limb, and some internal organs, I do see very strong concordance. So similarly, you can visualize individual genes, and you'll see a lot of interesting activities in brain tissues, and so this gene corresponds to the red blood cell production or erythropoiesis. And so at this particular stage of the developing mouse embryo, the liver is the red blood cell production organ. So that's why you see this strong enrichment of chromatin accessibility in the liver for the erythropoiesis. Okay, that makes perfect sense. All right. So what is much more powerful is if you have the single cell reference data, and you can sort of integrate the single cell reference data and our spatial sequence data by performing something called a co-embedding, and then you can transfer all different cell types you identify, you annotated using single cell reference data to spatial. By looking at, for example, here, if you see those red dots corresponding to our spatial ATAC pixels, if you see those red dots overlap with this orange cluster, or those red dots overlap with green cluster, and you know, okay, the dominant cell type in those red dots are, so in those red dots are determined by or dominated by this particular cell type, this cluster, this particular cell type. So using this kind of label transfer, co-embedding and label transfer, you can visualize individual cell types where there are, but in this case, what you see is not just the different cell types where they are, but whether or not they do show up as the epigenetically determined or epigenetically committed cell state, right? So I'm going to show you just a couple of those. For example, the urethral lineage, as I mentioned, is strongly enriched in embryonic mouse liver. You do see the label transfer, the co-embedding tells you they are right in here in the liver. And in the brain, you do see a lot of interesting neuronal cell lineages, accessory neurons, and some kind of premature, post-mitotic premature neurons, and OPCs, oligodendrocyte progenitor cells. So you can, after label transfer, you can visualize all different major neuronal cell types, but the take-home message is slightly different. Okay, this is not just about where the different cell types are. It is about whether or not at that particular stage the cells are or the epigenetic committed to become that cell type. Right? So since you are able to do spatial epigenome, spatial transcriptome, and the holy grail is, can you really perform both on the same tissue and the same cell, such that you can really link the gene expression to the epigenetic regulations, right? Turns out that's totally durable. And so I just show you maybe one or two examples. And this is, again, mouse embryo. And from this co-profiling data, but if you just take an RNA or take an epigenome to perform unsupervised clustering, you get a pretty nice clustering to delineate all major tissue types. And you can do co-embedding that worked out even better to resolve kind of more precise the tissue features. If you do kind of head-to-head comparison between the accessibility of the chromatin and the expression of the genes, in most of the cases, you do see pretty good concordance. And so now you may ask, right, since there's a pretty good concordance, but what's the added value? So that's a great question. So if you think you're just taking the epigenetics data, like either single cell or spatial ataxic to define the cell types, that does work in most of the cases, but that's underutilization of the very, very meaningful result. And what's the added value? So we believe probably most of the genes, okay, the chromatin opened up first and then the genes get expressed. That's true. But if you look at the gene by gene in this particular context, okay, the differentiation from radioglia to postmitotic premature neurons, and so you're going to see spatial, okay, that makes perfect sense. The radioglia lining around the ventricle, once they become more committed to neuronal lineage, they migrate into the parenchyma. But if you look at this differentiation process, and in terms of both RNA and the ATAC, in most of the cases, that's true. Okay, chromatin opened up and the genes are expressed. But some genes are interesting. Okay, for example, this group of genes, the chromatin opened up, that remain open, and the genes are expressed in the very beginning but quickly diminished. But what are those genes? When I ask my collaborator Gonzalo Castro Branco, and he found, wow, those are his favorite genes. They are non-neuronal cell lineage genes. They are actual oligodendrocyte lineage genes. So the implication is actually once the cells, the radioglia differentiate towards neuronal lineage, a subset of those still maintain the epigenetic, either epigenetic memory or epigenetic potential as a oligodendrocyte lineage. So that cannot be resolved by just one modality. Only if you have both, you can see, yeah, actually that's a two different epigenetic cell states that may still maintain the epigenetic potential to become oligodendrocytes. So you can perform this sort of shoulder time analysis based on epigenome or based on transcriptome. Based on RNA, you can see there's a monotonic differentiation from radioglia towards neuronal lineage. But in the ATAC data, you see, oh, there's a bifurcation. And there are two different states. One still maintain this epigenetic potential for oligodendrocyte lineages because they express the strong oligodendrocyte markers and their transcription factors as well. And so this is very interesting. So that means the cells upon external challenge, they may transdifferentiate, and it turns out recently my collaborators saw that does happen. And this is a complete new discovery. If you don't have the kind of dual modality multi-ohm spatial sequencer, you will not be able to discover that. All right, so we're able to also look at histone modification and the spatial RNA gene expression analysis on the same tissue, same section, spot by spot, pixel by pixel. And so I don't want to talk that much about the story, but just want to bring your attention to on the RNA data, right? So there are three different experiments, but I can see how consistent, at least these two are adjacent tissue sections. You see highly reproducible spatial transcriptome data from our assay. So maybe a few more minutes about what's in the future. I feel very humbled to speak about the future of the whole field, but I just want to show you what we're working on in my laboratory. I believe the power of the spatial omics technologies and where we should further cannot develop our technology to empower biological discovery. So as I said, I think the imaging based and the sequencing based spatial omics technologies emerged over the past 10 years, and they have their kind of pros and cons. The sequencing based approach allows you to do discovery, kind of genome scale, mechanistic discovery. So now the question, can we really combine both to get the best out of the both worlds, right? So in my lab, we are working on this high-plex immunofluorescence imaging, and the specific context is the serious senescence and the aging. I don't know if that's relevant in plant biology, but I believe the development and the aging, that's kind of ubiquitous in any living organism. And so we look at sort of all different cell types in human immune organs, such as lymph nodes across different age groups. And using high-plex imaging and integrate data with single-cell sequencing, we're able to identify all major cell types and go back to see where they are, right? So we basically build a cell atlas based on the high-plex immunofluorescent imaging. So now the question is, okay, from every single cell you see here, can you also get a genome-wide kind of discovery type data? So we found that it's totally possible using our in-tissue barcoding. So now we're able to get a data from the same tissue so for example, this particular region you see high P16, P21 senescent markers that are kind of senescent-like, but now you have unbiased genome-wide sequencing data, and now you can really discover what drives those cells to become senescent. Okay, they might become senescent through different biological pathways, but using the unbiased genome-wide either transcriptome or epigenome data, you are able to do that discovery, mechanistic discovery. Once you have the technology that allows you to get a unbiased genome-wide sequencing and high-plex immunofluorescent imaging from exactly the same tissue. So just some preliminary analysis. If you look at the cell type, right, without knowing the senescent or not, if you look at the transcriptome, and they all show as in the same cluster, right? But now using the senescent markers, you can separate them to two different regions to perform unsupervised sort of differential gene expression analysis. Now we do identify a lot of genes known to be associated with senescence, but we also see a lot of genes we never know. So now you can really discover biology from the same tissue and the same cell, so the information is extremely rich. So another, all right, so I think the background issue. So another powerful approach is we can further integrate spatial metabolic activity assay, showing here done by the microscopy technique called the super resolution stimuli Raman scattering in collaboration with Dr. Nietzsche and USSD. So from the same tissue and the same cell, now you can see the metabolic activity. Apologize for the description down here. You cannot see. This is a redox activity showing you there are more glycolic-like or there are more oxphos metabolic activity. We cannot know in a lysome the cells are more glycol versus oxphos. It is in a lysome, but you never know in the past which cells in the lysome upregulate their glycolic activity. Now, by merging data from multiple modalities on the same tissue section, you are able to answer that question and truly single cell resolution directly in the tissue. All right, so this is another example. So if you can resolve that in even the same type of cell hepatocytes, but in different zones of the human liver, all the way from the portal vein to central vein, you can see metabolic activity slightly different from central to portal vein, can directly visualize, and also every single cell you have high-plex immunofluorescent imaging and the whole transcriptome data. So I think I'm running out of the time. This has been published. This has been uploaded to bioRxiv, but I just want to emphasize the biology of the RNA molecule actually extremely rich. It's not just the gene expression, but the whole field of spatial transcriptomics to some degree is equal to spatial gene expressions. We're asking, can we really understand the biology of every single RNA molecule directly in the tissue across the entire life cycle, like from the transcription to maturation, splicing on a different isoform and the translation degradation. So can we look at those? And also not just the messenger RNAs, but on a regulatory sort of the non-coding RNAs. So it turns out that's totally possible, similar to the talk given by Stefania. Using the polyadenylation approach, you can probe all different RNA species, which is what we didn't anticipate is worked out so well. It worked out way better than fresh frozen samples. And so now in a FFP tissue specimens, I don't know plant biologists whether or not you guys do FFP, but turns out this paraffin fixing actually help preserve the RNA molecules, in particular the small RNA molecules, non-coding RNA molecules. Okay, and the data is amazing. Just bear with me. If you want to know more details, you can go to check out this bioRxiv paper. So I'm gonna just finish up my talk uh, uh, by showing you what we have been working on and in terms of technologies. And our own research interests are largely in two different directions. I think one is the immune system, immunobiology, and also some neuroscience. But the key takeaways, I really think the whole field is really like a family nowadays, and we all work together. Together is better, and the team science, teamwork really kind of advance science and one day help promote humanity and generate societal impact. So I would like to thank people in my laboratory, current and former, and amazing collaborators, funding support. And thank you so much for your attention. It's such a great honor and a privilege to be speaking to the plant biology community, and I'm happy to answer a few questions if time permits.
All right, thank you for really informative and inspiring talk. And we have a couple minutes till the end of the session. So I think it's a good opportunity to revisit one question asked to Stefania. So the question was about the possibility of spatial genome sequencing, spatial DNA sequencing. And I guess your spatial ataxic approach could be applied to, for example, spatially map SNPs and structural variants in, say, cancer cells and so on. So could you comment on that possibility?
Yeah, I would like to kind of point your attention to the paper already published by Fei Chen and the Broad
Yeah.
Institute. They did a slide DNA-seq. So basically, it is possible to do spatial genome sequencing. Turns out that's easier, honestly. That's slightly easier. And the epigenome is harder because it's not about whether or not the DNA molecules are still there. Okay, they're not degraded, they're well preserved. It's also about whether or not the DNA protein interaction is also well preserved in your tissue. So that turns out to be harder. So I would say spatial DNA sequencing totally durable. That hasn't been our focus, but my former postdoc is working on that right now in his independent laboratory, and also Fei Chen already demonstrated. But of course, the question too is, you can do it, but how good you can you can get your data, right? So I don't have the spatial epigenome data in my lab, but from the transcriptome, we can infer, or you can kind of detect a lot of kind of genetic variants. I probably don't want to call them mutations, but they're kind of like a mutations on a SNPs but maybe I should not call SNP. I call it single nucleotide variants we detect in the RNA in the FFP samples. Yeah, somehow the data is pretty unbelievable, and from the FFP fully fixed tissue samples that worked out way better than fresh frozen specimens.
Mm. Okay, so one last question. It's a kind of big question from Xiaohui Li, so who asks, what will be the next giant leap towards spatial transcriptomics or spatial omics in live tissue to get some temporal dynamics?
Yeah, I think my own interest for many years has been kind of how to make the technology compatible with clinical practice, right? Clinical pathology practice. So in that case, they have their standard tissue procurement processing. I kind of try not to change anything on their side. And for life, I would say maybe the answer twofold. It depends on how you define life, right? You can do maybe in vivo CRISPR and lineage tracking stuff. You can record something associated with the molecular biology when the cell is still alive. But afterwards, you take one snapshot, spatial omics sequencing, but you can go back to see what those cells have done by reading out the recorded information. So that is possible. That's one way to do. Also some metabolic labeling of the RNA, you know, which ones are newly synthesized, which ones are old. That gives you some sort of temporal life cell aspect. So, but the assay itself is not done on live cell. But I know one of my friends, Bo Huang at a UCSD, UCSF, and he's developing all those kind of transfection construct, and you can actually do extremely high plex, and his goal is to really map out all the kind of protein-protein interaction, the genetic elements in a live cell. But I think it's probably hard to go to fuller genome scale in that case. But he can go much higher plex. And once you get enough plex, actually you do get some level unbiased view or global view. So that's still kind of very valuable. Yeah.